this video, we're going to talk about proteins and how the structure of the protein determines its function and how the structure is determined. Proteins have very different functions in the cell. They really can do pretty much anything the cell needs. So proteins can do catalysis or catalyze reactions. Those are called enzymes. They can do transport, so they can carry, for example, oxygen as hemoglobin. They can act as defense or protection. So, for example, our nails are made of keratin, which is a protein, and that serves as defense or protection from injury. They can work as support, building structures for the cell, for example. So actin filaments that help support the cell are made of proteins. For motion, proteins that make our muscles, they help us move, and they also help the cells move. They can help us regulation, so some proteins are hormones, and they can also be used as storage of energy. And what determines the function of a protein is its structure. So the structure of the protein tells us what is the function for that protein. And all these possibilities for proteins come from the way proteins are built. So proteins are made of amino acids, and there are 20 amino acids, but they have very different properties. So we have amino acids that are non-polar, so like this group. We have polar amino acids. They don't have a particular charge, so like water that is not charged but is still polar, as they have simply an uneven distribution of charges. Others that are charged amino acids, and they can be positively charged or negatively charged. And we have aromatic amino acids and also special kinds of amino acids. What gives them the identity is what we call the R group. Each amino acid has a different R group, but they all have the common amino and the common acid group. So this is the organic acid group, and this is the amino group. And every amino acid has that. And what gives them the identity is the R group, which is highlighted in the white boxes. Then amino acids are linked together by what we call peptide bonds. So this is a bond between the acid group of one amino acid and the amino group of the next one that results in a dehydrating reaction. So water is released, and now we have dipeptide, which means a peptide made of two amino acids. So peptides is the name for a chain of amino acids. And our genes are the ones that determine the order of the amino acids. So proteins are all coded by our genes. All the proteins we produce them because we have the instructions to make them in the DNA. However, the DNA is only coding for the order of the amino acids. Each group of 3 DNA nucleotides is coding for a particular amino acid. The sequence of the DNA is determining the sequence of the amino acids in a protein. However, the sequence of the amino acids is only the beginning of the structure of a protein. The amino acid order or the amino acid sequence is what we call the primary structure. So this is the very beginning in the making of a protein. Once you have the primary structure, then the protein starts to fold. The next step is folding into the secondary structure. Proteins fold into secondary structure by making these beta sheets, or they can form alpha helixes. So these are the two main secondary structures that proteins form. And these secondary structures are formed by interactions between the amino group of one amino acid and the acid group of another one, forming hydrogen bonds. So hydrogen bonds is what is holding together the secondary structure of a protein. After the secondary structure is folded, the most complex form of the protein folding is the tertiary structure. So imagine the protein goes through several foldings. It's like knitting a sweater, and the first structure you will have the thread, and then you look at the thread. Threads are often twisted, so that would be the secondary structure. And then how you actually knit it into either a sweater or a hat, that would be the tertiary structure. So there are, again, many possibilities for a protein to fold onto its tertiary structure. And the tertiary structure is determined by interactions between the R groups. Every amino acid has a different R group, so the possibilities on how they interact are very wide. So there are multiple forms in which the protein can form, but only one form is suitable. So the type of interactions these R groups can have: they can form hydrogen bonds, they can form disulfide bridges, so these are proteins only for amino acids that have sulfide. They can form ionic bonds, so this is a pretty strong interaction, or they also can form van der Waals attractions, which are interactions between hydrophobic amino acids. Phobic amino acids, they want to shield themselves from the hydrophilic environment or the water around them, so they will form these hydrophobic exclusion areas in which they're all folded onto themselves. So all these interactions result in the tertiary structure of the protein. Since there are so many options that a protein can fold, most proteins need assistance from other proteins called chaperone proteins in order to fold correctly. So chaperone proteins help the peptide as it's forming acquire the structure for it to function correctly. Some proteins might then, after they're folded into their tertiary structure, they might come together with other peptides, so these are other amino acid sequences, and form a quaternary structure. So not all proteins form this. This is just separate proteins coming together and forming a larger protein that is called the quaternary structure. So just like we said how important the structure of the protein is for its function, the same is true if the structure is lost. And that's what we call denaturation. So denaturing is when the protein unfolds. This happens for mainly two reasons. Either because of changes in pH. So proteins, they have a very specific range of the pH that they can tolerate, and anything outside that will cause them to denature, or if the protein is exposed to very high temperatures. Now notice that what makes a protein denature depends on what the protein is meant to be. So for example, proteins in our stomach are used to be in very low pH or in a very acidic pH. For them to denature, it would take an even lower pH than for a protein in other parts of our body that are used to a more neutral pH. Actually, the acid in our stomach helps us in digestion because it denatures the proteins that we're eating. So most proteins will be denatured in that acid, except for the proteins that are produced in the stomach, which are meant to tolerate that acidity. The same thing with high temperature. So most of our proteins will denature if they take temperatures higher than what we're used to. But there are some bacteria that live at very high temperature, and their proteins are made so that they can tolerate that temperature. So there is no specific pH or a specific temperature that affects all proteins, but every protein has its own range, and if taken outside of that range, it will denature. And when a protein denatures, it will lose its tertiary structure that gives it its function. And this will change the properties. So you can see this when you're cooking an egg. It goes from being a runny egg to being almost solid. And this is happening because the proteins are denaturing from the temperature. There are diseases that can be also caused by proteins folding in a different way. So in this case, they're not necessarily denaturing, but they're acquiring a different structure. This is the case of mad cow disease that causes these nodules in the brain, and it results in death in most cases because of impaired brain function. The natural protein has this way of folding, and that is the misfolded version of the same protein, what is called the prion. So the same amino acid sequence, nothing in the genes has changed for this protein. Once this normally folded protein gets in contact with the prion version, it will misfold and it will turn into a prion itself. And when they misfold, they aggregate and they stack up together and they form these big clumps of proteins, and those are those nodules that we see here that are the ones causing the problems in the brain tissue. In this case, just contact with a misfolded protein causes a normally folded protein to misfold, and then that would start to form these aggregations. Prions are acquired by eating tissue that has these prions, and then they will find their way into our normally folded proteins and cause them to misfold. The most common way in which proteins will misfold is if their primary structure is not correct. This is what happens when you have mutations. Mutation is just a change in the DNA. If that change in the DNA causes a change in the amino acid coded, that could result in a change in the function of the protein. So it depends on where the amino acid change happens. So if it's in an area that is very important for the function of the protein, or if it's in an area that is not so important, and also what type of change. So if you're changing one amino acid for a similar one, let's say a polar for another polar that are more or less similar, then that might not be such a big deal. But if you're changing the amino acid in an important place and you're changing it for one that is very different, that will result in a huge change in the function of the protein. That's the case of sickle cell anemia. In sickle cell anemia, we have the primary structure of the normal protein. This is just a little bit. These are only 7 amino acids in a very long protein. And this is the sickle cell version in the bottom. And the only difference is this amino acid here The rest of the code, which is not shown here, is the same, but this amino acid in the normal protein is a glutamate, which is a polar amino acid, and is changed for a valine, which is a non-polar amino acid. So this is a very different type of amino acid, and it happens to be in a very important part. So that when here we have the primary structure has been affected, when the protein falls into the secondary and tertiary structure, the one with the sickle cell gene is with the valine instead is going to form a very different structure. The tertiary structure is affected. This protein comes and joins other proteins, forming a quaternary structure. When that happens, that is also affected in the sickle cell. The main difference here is that this new structure causes this protein to aggregate and form these long threads or long fibers, while normally hemoglobin will not aggregate. Once it reaches this quaternary structure, it would just stay independent. But here, since they form these aggregates, change the shape of the red blood cells. So here we have normal red blood cells with normal hemoglobin, that is the globular form, so it's not attaching to each other. And here the sickle cells, they have these long strands, long fibers of hemoglobin that is attaching to each other, and that stretches the cells and changes the shape so that the cells can no longer flow easily through the vessels.